Welcome. It's a pleasure to have our speaker today, Professor Jonathan Liu from University of Washington here. He's the director of the Molecular Biophotonics Laboratory. It's a special pleasure for me. I work in the same area that he does, and I've watched him progress through his career over many years now. It's been just a lot of fun to see the creativity and the innovation of what he's been working on. He started his academic life at Princeton University, where he did mechanical engineering bachelor's, graduating in '99 with an award as the top graduate in mechanical engineering at Princeton. He then went on to Stanford University, completing a master's in 2000 and a PhD in 2005, and was awarded the National Science Foundation Graduate Research Fellowship. He then became a postdoctoral fellow in electrical engineering in the Ginson Labs and at the molecular imaging program at Stanford for 4 years, where he received the Canary Foundation and American Cancer Society postdoctoral fellowship. He was then appointed as an instructor at Stanford University School of Medicine and received a K99R00 fellowship from the National Institute of Health. He then moved across the country and joined as an assistant professor at SUNY Stony Brook, and was there for 4 years, from 2010 to 2014. And then he moved back across the country, and he's now an associate professor of mechanical engineering at the University of Washington in Seattle. His laboratory works in molecular biophotonics, developing optical strategies for improving the diagnosis and treatment of diseases. He's published over 70 papers by my count, and I'm really looking forward to seeing what he presents to us today. So please welcome him.
All right, thanks a lot, Brian, for the introduction. Can people in the back hear me okay? Is this okay? All right, great. So I'll be talking about my lab's work in molecular biophotonics for early cancer detection and surgical guidance today. So I'm in mechanical engineering. That's where I got my degrees, but I think of myself more as a biomedical engineer, sort of multidisciplinary skill sets. So what we do is translational research, meaning we're trying to translate technologies from a laboratory setting into a clinical setting in biomedical optics. And mainly what we're trying to do is to improve disease detection as well as treatment of cancers. And so my background as a mechanical engineer started actually in the field of combustion. My PhD was in the area of combustion diagnostics, building optical sensors for engines. So we did a lot of optical design, mechanical design of hardware, instrumentation, so a little bit of electrical work. We use things like MEMS mirrors. I'll show you an example of that. And molecular imaging refers to the imaging of protein biomarkers or genetic biomarkers that are overexpressed by cancers, which involves some medicinal chemistry, nanoparticles, contrast agents, image processing to make those images quantitative, and working with surgeons and other types of clinicians, pathologists, using animal models as well as ultimately trying to translate these technologies into patient care. So I'll be talking about three main areas in my lab. The first area is what I'll call point-of-care microscopy for in vivo cancer detection and treatment. These are handheld microscopes or endoscopic microscopes that can be placed into patients. I'll talk about the application of brain tumor resection, but we're also trying to use this for early cancer detection in the oral cavity. Another area I'll touch on briefly is multiplexed molecular imaging with these surface-enhanced Raman scattering nanoparticles, and I'll explain why we're using those nanoparticles in that part of the talk. Focus primarily on guiding breast cancer surgeries to remove breast cancers, as well as some previous work trying to do endoscopy of molecular biomarkers in the esophagus. And then finally, an exciting area I'm talking about is all unpublished at this point. It's a new area in my lab for rapid, non-destructive, slide-free molecular 3D pathology of tissue specimens. This could be used for surgical guidance as well as for diagnosis of tumors and guiding treatments using core needle biopsies and assessing them. So that's an exciting new area that I'll try to spend quite a bit of time on towards the end of my talk. So I'll start with a motivation for the in vivo microscopy work where we're trying to build a handheld microscope. So many of you probably know that brain tumor patient outcomes correlate with the extent of resection, meaning we want to be able to remove as much of the tumor as possible in the patient because studies have shown that that improves their outcomes. But surgery in the brain is highly qualitative and subjective. The surgeon is basically looking at different colors and textures and feeling for the tumor in certain cases. So it's not a very quantitative or accurate way of delineating where the tumor ends when the normal tissue begins. And for a lot of these brain tumors, they're very diffuse. So there is no clean-cut margin. It's not like a well-circumscribed solid tumor. It's actually diffuse and infiltrative. Now, for disease diagnosis for probably 100 years or more, the gold standard is based on something called histopathology, which is taking tissues out of a patient and then preparing microscope slides, cutting the tissues into very thin sections, staining them, and viewing them under a large microscope. But there are limitations. This is obviously invasive. You have to take the tissue out of the patient before you know what it is, which is not something you want to do in the brain if you don't have to. Time-consuming and costly. Generally takes a few days for this procedure to be done. And there's sampling errors. They're only preparing a few microscopically thin tissue sections on microscope slides. They're not actually imaging through the entire piece of tissue that they extract from the patient. So it's less than 1% generally of the tissue they're actually inspecting under a microscope when they're doing pathology. So there's clearly a clinical need for both intraoperative guidance in the form of wide field imaging, being able to look at the entire tumor cavity through fluorescence image guidance, something that's been done here a lot, as well as intraoperative MRI or intraoperative CT. But there's also a clinical need, we believe, for intraoperative high-resolution optical biopsy using a variety of different approaches where you can see down to the cellular and subcellular level. And the reason for that is because the gold standard for hundreds of years has been based on microscopy, being able to see cellular nuclei and observe the cellular and glandular architecture. That's the most reliable way to date of being able to diagnose what a tissue is. So we're working on what's called a confocal microscope, and I'll go over kind of the conventional confocal microscope architecture known as a single-axis confocal architecture before talking about our technology over the last few years, which is the dual-axis approach. So with a standard confocal microscope, what the goal is to be able to image beneath the surface of the tissue in 3 dimensions at the microscopic scale. And the way that you're able to image beneath the surface is to filter out the background light, because the light, when it enters tissue, scatters like crazy. It's like having headlights in a fog. You have a lot of glare, you have a lot of back reflection, back scattering. And so that's a problem if you were trying to image cells underneath the surface very clearly. The way that a single-axis confocal works is by taking a point source of light. In our case, we use a single-mode optical fiber, and we focus that light with what we call a high numerical aperture objective. That means a lens that's focusing the light with a very sharp angle into the tissue. So we have a well-confined focal spot within the tissue, and the light that's generated from that tissue, whether it's reflected light or fluorescence light, can be collected back into the same pinhole or some other pinhole. And the point is that only the light from the focus is able to propagate very efficiently back into this pinhole, because you're mapping this point back to this point here. But the light everywhere else, the out-of-focus light, is largely rejected by that pinhole. For example, light up here is going to focus above the pinhole. Light over here is going to focus to the side of the pinhole. Only the light from the focus makes it efficiently back through that spatial filter. So that's the way that a confocal microscope is able to reject the out-of-focus light, the multiply scattered light, and to generate a clear image beneath the surface if you can scan that point by point in 2 dimensions to create a 2-dimensional image. But it's not a perfect optical sectioning device. There's still a lot of background noise. There's still a chance that some of this out-of-focus light or scattered light can make it back into that pinhole. And we've done a lot of theory, diffraction theory modeling and Monte Carlo simulations, which I don't have the time to show. But it's very clear experimentally and in simulations that there's still a limitation to this approach. And we can typically only image maybe 100 microns deep with this confocal architecture. Now, with the dual axis architecture, instead of using a single path arrangement, we illuminate the spot from the side. So we're sending in light off axis through this low NA beam, meaning a weakly focused beam. Now, for those of you in the optics field, you know that a low NA beam means that you have a fairly weak focus, and it's not a well-defined focal spot. And so that's poor. That's bad if you want to have very high resolution to image small structures. But if you intersect this illumination and collection beam, the focal volume of this microscope is actually defined by that intersection of those two beams, which is still fairly well confined in space. And as you can see, with this arrangement, you've separated the illumination light from the collection light. We've shown through a lot of modeling, again, that I'm not showing, that we're able to reject this out-of-focus light much more efficiently with this arrangement. It's sort of intuitive that you're separating the scattered illumination light from the collection beam, so there's less chance that the scattered light can make it into the collection pinhole. So as a result, we can image more deeply. We also get a longer working distance here because of these low NA lenses, which for miniaturization purposes is helpful. We can actually put in a tiny mirror to scan the beams after they're being focused, and I'll talk about that design in a second. These are just some of the examples of images we can obtain with a tabletop dual-axis confocal microscope. This is human esophagus tissue. Here's a vertical section, so this is the surface of the tissue, and without cutting the tissue, we're using light to image beneath the surface, and we can clearly see the cells. In this case, this tissue was stained with a fluorophore that tends to highlight the cell membranes. In terms of building a miniature device, this was our first generation device. Actually, back when I was at Stanford, we built this fiber-coupled laser going into a small handheld unit where we collimate the beam, both for the collection and the illumination beam paths. And there's a tiny MEMS mirror here that was developed at Stanford, which focuses the light to this point here. And then that's relayed forward through this relay optic, a needle lens that brings the beams into the patient at this side. The focusing for this device was done with a parabolic dish, similar to some telescopes, Cassegrainian telescopes, where as long as you have parallel and collimated light hitting the parabola, it ensures that these two beams will intersect and focus at the same point, which is at the focal plane. So the MEMS mirror steers that point so that you can image in 2 dimensions. And initially, we were doing a lot of imaging in a preclinical animal model. This is a spontaneous model of the most common form of pediatric brain tumor known as medulloblastoma. It's a patched p53 genetic knockout mouse that develops these tumors spontaneously and expresses a green fluorescent protein through a developmental gene that is overexpressed by these tumors. So this worked pretty well for imaging mice, but it had some limitations. One was the spatial resolution wasn't great. The axial resolution was about 9 microns, lateral resolution 3 microns. So if you want to be able to image subcellular structures, you'd like to be able to do a little better than this. The other problem for clinical use was it had a slow frame rate. This was a point scanning device, so we had to raster scan this point in 2 dimensions to create a 2-dimensional image. Hold on one second. Should pop up. Oh, interesting. Let me try unplugging this. Maybe that'll work. Okay, here we go. Okay, so as I was mentioning, the slow frame rate is a problem clinically because you get motion artifacts, basically. If you're only imaging at about 4 frames per second and the surgeon's hand is jittering, you're going to have a lot of blur in your image. So ideally, we want a faster frame rate so that we can do what's like high-speed photography, freeze the motion and get a clearer image. That's challenging with a point scan microscope where you're collecting the image point by point. But another approach to doing this is to focus the light into a focal line instead of a point, and scanning that line in only one direction to create a 2-dimensional image. So we have a detector on this side that images that focal line. We collect all of those pixels all at once, and by scanning that line in one direction, we can collect a two-dimensional image. The trade-off is you lose confocality along that line. It's not as good of a spatial filter compared to the point-scanned dual axis. And that's reflected in these images. You can see that the imaging depth of a point-scan microscope is deeper. But if you're interested in just imaging at the surface, you get comparable performance. And so with the neurosurgeons, they were really just interested in looking at the tissue that they could see in a tumor cavity at the surface and knowing if that was tumor or not. And so we felt like this would be a good approach. These are just some example images taken from a tabletop version of a line scan dual axis confocal microscope that we used at first. This shows ability to image at 15 frames per second. We can actually increase that to 30 frames per second if we reduce the field of view a little bit. And we're also able to get decent signal at this frame rate, which is important. If you're imaging that quickly, sometimes you wonder if you can get the sensitivity. And in this case, we've applied an FDA-approved agent, methylene blue, onto the tongue of a mouse, and we're able to see the microscopic structures. So that gave us confidence that we could build a miniature device and try to get that into patients. This is what the device looks like in a schematic form. There's 3 main modules, a detector, a linear detector unit that images that focal line, the main body where we do a lot of the alignment, and there's the MEMS mirror, which I'll show in the next slide in more detail. Focusing optics, alignment prisms allow us to bring these two illumination and collection beams exactly into focus with each other. And then in collaboration with a company in New York, we developed a custom objective lens to magnify the beam so that we can achieve high resolution down to about 1 micron in the lateral direction and 2 microns in the axial direction. This is a photograph of what the device looks like. This MEMS chip is developed by a company in near Berkeley, California. It measures about 8 millimeters by 6 millimeters. And it scans, it only has to scan in one direction in this case. It's a slow mirror, but for us it's fine because we only need to scan that line in one dimension to create a two-dimensional image. This is the custom machine part that holds a lot of the optics, custom objective lens. And here are some of the images that we were able to obtain. Right now it's been done in mouse tissues, looking at the tongue, kidney, colon, but you can clearly see we have nuclear resolution with this methylene blue dye, and we can get comparable information to the H&E pathology that pathologists are used to seeing. So in terms of next steps, if I back up, we know that wide field fluorescence image-guided surgery is effective. There have been studies in Europe and in the States, as well as here, showing that this compound known as 5-ALA, 5-aminolevulinic acid, when the patients drink it 3 or 4 hours prior to surgery, their tumor cells metabolize this dark chemical into a fluorescent porphyrin, PP9. And so that is a metabolic marker of the tumor cells, which convert molecule, and it's been used to guide surgeries. It has this pink fluorescence that you can view under a surgical fluorescence microscope, and it's been shown to help with high-grade glioma patients. The problem is image intensity is still subjective. You're looking at different shades of color and different brightnesses, and at the margins, as I mentioned, these tumors are diffuse, so the tumor cell density is kind of fading away at the margins. There's no real good way to quantify that tumor burden. So it is a challenge, and especially at the margins as well as low-grade gliomas just don't produce a lot of this PP9. These techniques often don't provide enough sensitivity to see any signal. The advantage of microscopy at high resolution is that we can actually see at the subcellular level these sparse expressions of PP9. They're like stars in the sky, really. And we believe those are from the mitochondria, where there are reports showing that that's where the conversion of 5-ALA to PP9 occurs. And so we've shown that in collaboration with our neurosurgeon collaborator, that even when wide field microscopy lacks sensitivity, we can still see the small spots of PP9 under a high-power microscope. And hopefully we can use that to quantify the PP9 expression and to allow surgeons to optimize their surgeries based on a more objective and quantitative metric. So that's kind of the direction we're moving in with our neurosurgeon collaborators. Okay, so I'll switch gears. I'll just maybe spend a few seconds talking about our oral cancer project. We're actually trying to get our device into the clinic at Memorial Sloan Kettering Cancer Center in New York in the next few months. And we're going to start with oral cancer detection. The problem there is there's a lot of lesions in our mouths that look like cancer, but very few of them are. And so dentists and clinicians are very hesitant to biopsy these lesions. Most of them are normal and benign. But a few of them, if they are cancer, you want to catch them at the earliest stage where they're treatable. So if we had a handheld device, non-invasive, you don't have to take out tissues from the mouth. If we can place that into the oral cavity and inspect these lesions, it may be a way to catch these cancers much more early before they progress. So the next topic I'll be speaking about is this multiplex molecular imaging with Sears nanoparticles. And so the clinical problem here is breast conserving surgery. And I think many of you know this is one of the biggest problems in the world. Many of these procedures are done. But the problem is that 20 to 50% of these patients require multiple rounds of surgery to remove all traces of the cancer. So there's no good way to image these tissues and to decide if there's tumor left at the surgical margin. If there's tumor left at that surgical margin, that means there's still tumor left in the patient, and you want to keep cutting, ideally. So what we proposed to do was to use some molecular imaging, that is the imaging of these proteins that are overexpressed in these tumors to detect the tumor. The challenge is that these molecular profiles are very heterogeneous. So different patients express different protein biomarkers, different genetics. And so if you want to be able to detect tumors in many patients, ideally you want to target a panel of different biomarkers and be able to image simultaneously 5 or 6 or maybe even 10 different biomarkers. The way we do that is with these surface-enhanced Raman scattering nanoparticles, which you can think of as barcodes. Each of these nanoparticle flavors emits a characteristic fingerprint spectra. So if we shine one color of laser light at 785 nanometers, each of these nanoparticles will emit a characteristic set of colors back at the device that we're imaging these nanoparticles with. And with a spectrometer unit, we can disperse those colors and figure out through their spectra what nanoparticle is in that mixture. So I'll talk a little bit more about that in a few slides. But to make this work for molecular imaging, you first have to target the different flavors of nanoparticles to different biomarkers. So I won't go over the complexity of the chemistry, but in a nutshell, what we're trying to do is to conjugate monoclonal antibodies to these nanoparticles, both targeted as well as an isotype control, a negative control antibody, which I'll talk about a little bit later, the purpose of that. So this is work that we did in collaboration with Brian Wilson, who was Brian's PhD advisor, is that right? So we do topical delivery because we're looking at excised tissues. We're not imaging on the patient, but taking the tissues from surgery and immediately imaging it at the patient bedside or in a frozen pathology suite next to the operating room. So there's a few advantages of topical delivery, as many people here know, because they do that as well. One is reduced toxicity and regulatory issues. Where we don't have to systemically deliver a chemical into the patient. And it's potentially much more rapid. You don't have to wait hours or days for that chemical to circulate through the body. We can just apply it on this freshly excised tissue. Now, for surface imaging, the penetration depth of these fairly large nanoparticles is limited to maybe only a cell layer or 2 deep. But for us, that's actually a good thing because it allows us to wash out the unbound nanoparticles very quickly. We found that if they go in too deep and they diffuse too deeply, they actually get trapped. And so they don't wash out and you don't get the good contrast. This is an example of an image showing that they're really only penetrating about 10 microns deep, about a single cell layer deep on fresh tissues. There are challenges for topical delivery. One is rapidly achieving sufficient binding of the nanoparticles to their protein targets, and as well as getting enough contrast between tumor and normal based on the biomarker expression. There's also always a lot of non-specific binding. I mentioned trapping and diffusion. Being unable to wash out the unbound nanoparticles can be a problem. And so we do some image processing approaches, which I'll talk about. And similar to some of the work that's being done here with Scott Davis and Brian Pogue and Summer Gibbs, where they've tried to do similar approaches with dual fluorescent probes to try to quantify the specific versus non-specific staining of these topically applied probes. So the way we do it is we first apply a one-to-one mixture of our targeted and untargeted nanoparticles. So it's a one-to-one mixture. It goes onto the tissue, and we let it sit for maybe 5 minutes. Then we rinse off. And for the normal tissue, we will always have a ratio of 1 because there's no biomarker of interest. All the nanoparticles stick the same. There is nonspecific binding, but it's not a differential binding. But with the diseased tissue, you'll have an elevated ratio of your targeted versus your non-targeted nanoparticle because it's binding more to the biomarker of interest. So this allows us to have unambiguous molecular imaging of these biomarkers. And without this ratiometric approach, it's very ambiguous, as I'll show you in a second. This is the device. It's a raster scanning stage, so it's a point-by-point measurement device where we illuminate with 785 nanometers through a fiber and collect the Raman light through a multimode fiber bundle into a spectrometer. The light gets dispersed by diffraction grating, and then we get a spectral pattern on our CCD camera. We use some demultiplexing. Once we have the combined signal from many nanoparticles, we can extract how much of each nanoparticle is in that mixture through some linear least squares demultiplexing. So basically, if we know what each nanoparticle produces in terms of its spectra, and if we measure the tissue background components, we can back out how much of each nanoparticle is in the mixture on the tissue. So here's an example which we've topically applied onto a xenograft tumor on a mouse, a 3-flavor mixture. So 2 targeted nanoparticles and 1 untargeted nanoparticle in 5 minutes, rinsed for just 20 seconds. It's a very rapid rinse. And then point-by-point measurements at 0.1 seconds per point. So if you look at the absolute concentration of nanoparticles, it's kind of interesting. The normal tissue actually picks up more absolute concentration of nanoparticles. And that's because these xenografts tend to be fairly dense and form a barrier to these nanoparticles getting in. And the normal tissue is a little more porous. But if you look at the ratio between the targeted particles, including an EGFR targeted and a HER2 targeted nanoparticle, compared to the isotype control, immediately you see that the ratio is elevated for these tumors that express EGFR in this case, and this tumor that expresses HER2. So it's very different from the normal tissue. So it shows you the utility of the ratiometric approach. We started imaging some of these xenografts point by point as well. Again, the surrounding normal tissue actually displays more signal from the nanoparticles, but if you take the ratio of the targeted and untargeted, you can clearly see this is the tumor that highly expresses EGFR. This is the tumor xenograft that highly expresses HER2. And you can kind of do multiplex imaging of these different biomarkers. So it allows us to do multiplex imaging of superficial tumors through rapid topical application, just 5 minutes of staining and imaging. So then we moved into the clinical specimens. These are breast tissues that either express HER2 or don't. In this case, again, the absolute concentration of nanoparticles is very ambiguous. But if you look at the ratio between the HER2 and isotype nanoparticle, you see it agrees very well with the immunohistochemistry. This is sort of the gold standard that pathologists look at, and you can see that the tumor region lights up. And for HER2 negative tumor, you don't have any accumulation of the preferential accumulation of the nanoparticle. Since then, we've looked at many different biomarkers in fresh human breast specimens, including EGFR, HER2, some CD markers, CD44, CD24. And we've shown that they're able to reliably differentiate between positive, for example, CD44 positive versus CD44 negative tumors. And so we're in the middle of a clinical study to assess the sensitivity and specificity for tumor detection based on molecular imaging of these markers, and hope to get that paper written pretty soon. Before I end, let me just mention A lot of the success of this has to deal with how we stain the tissues. Turns out we have to design a system to mechanically assist the staining. And that's because these nanoparticles are relatively large, about 120 nanometers large. So the convection is actually fairly poor near the tissue surface. For those of you who have taken fluid mechanics, you know that there's a boundary layer, a low velocity boundary layer near the tissue surface or any interface. And so that restricts the motion of the nanoparticles so that they don't efficiently find their targets. We found that by dipping the tissue into and out of this solution, we're able to really enhance the efficiency of the staining as well as the rinsing of the nanoparticles. Another area we're looking into, which is very familiar with a lot of you here, is kinetic modeling. This is work done with Ken Tishar, who used to be a postdoc with Brian, now at Illinois Institute of Technology. But trying to look at the kinetics of the staining and the rinsing off of these nanoparticles through some compartmental modeling, which should allow us to more accurately quantify what's called a binding potential, which is, in essence, it's the receptor density. We want to know how many proteins are there per cell. And by doing these kinetic experiments where we image at multiple time points and watch the binding and the release of the nanoparticles, we can better quantify that value. And I'll just race through one more application of this. When we first started, we were interested in endoscopy. And this is something that some folks at Stanford are very interested in, being able to do endoscopic imaging with these nanoparticles to look for cancers of the esophagus or colon. We didn't want to go through FDA approval. They're doing that at Stanford, still slogging through that. When I started my faculty position at Stony Brook, I wanted to get into a realistic model of this. So I started with an animal model, a rat model of esophageal cancer, and built a tiny rat endoscope that scans the lumen of the esophagus in a spiral pattern back and forth. And as it gets pulled back, it images the lumen of the esophagus. So we show that this works pretty well. When we irrigate the esophagus with the nanoparticles and insert this probe through a glass guide tube, we're able to image these tumor phantoms. This is showing, again, that without ratiometric imaging, it's a very messy image. But as soon as you take the ratio, you can spot where the tumors are pretty reliably. We also did some in vivo imaging. We implanted orthotopically 3 different tumor xenografts in a mouse, actually a rat esophagus in this case, inserted a glass guide tube and inserted our scanning device. And we were able to pick out the high EGFR expressing tumor in this case, the high HER2 expressing tumor in this case. And if we compare our ratiometric measurements to flow cytometry, we have very good agreement between flow cytometry of the cell lines that were used to develop these xenografts. So it seems to be quantitatively accurate and comparable to flow cytometry. Okay, so let me make sure I'm okay on time. All right, good. So last topic, which is very exciting to me because it's new in our lab, is this topic of rapid, non-destructive, slide-free molecular 3D pathology. It's a lot of words. I'll explain what they mean in just a second. So the motivation for this is that pathology, as I mentioned in the beginning of my talk, has remained virtually unchanged for about a century. This is the gold standard for disease detection. It involves taking tissues out of a patient, chemically processing the tissue, for example, fixing it, and then dehydrating the tissue, embedding it in a wax substrate, or freezing the tissue and putting it in a freezing substrate, sectioning it with a very sharp blade, mounting those tissues onto glass slides, staining them, putting a cover slip on it, imaging under a microscope. It takes days to do this typically. This is expensive, time-consuming, it's destructive, and there's major sampling errors because you only can cut a few sections onto microscope slides. You can't process through the whole tissue. That would take way too many resources. So the goal is, can we do non-destructive, slide-free digital pathology? So this whole idea of 3D digital pathology has been around for a while, but people have been doing it by scanning microscope slides, glass slides, and scanning them with a device. And that's never going to be really feasible in terms of economics and time because the slide production process is just too onerous. And so if we can get around the slide making process that could help a lot. So the way this works in our current form is we first do some optical clearing if we're working with biopsies. This is a chemical process that makes the tissue more transparent so you can image more deeply. Then we can stain the tissue. Actually, the optical clearing procedure, which involves the electrophoresis, assists with the staining, and we can stain very deeply into the tissue. And then we want to image the tissue in its uncut state, non-destructively. So the question is, which microscope do we want to use for imaging? And there's a lot of different three-dimensional microscopy technologies that have been out there. But one major challenge that we identified early on was that with a conventional microscope, if you want high resolution, you typically have a very shallow depth of focus. Anyone that's done microscopy knows this. At the high power setting of your microscope, the focus is very hard to find. And if you have an irregular tissue surface, as any surgical specimen is, you can't image that surface very easily. You have to do elaborate tissue flattening, or you have to image at many different depths, which takes a lot of time for a confocal microscope or a two-photon microscope. Light sheet microscopy is becoming a hot area, and it's been for many years for developmental biology, but it takes a light sheet, basically a sheet of light that cuts into the tissue at an angle, and we image it at a right angle so that we collect a full two-dimensional image all at once. And by scanning the tissue laterally, we can collect a three-dimensional volume of data by simply scanning the tissue in one dimension. So it's a rapid way of performing 3D imaging, and the assumption is that this tissue surface is contained somewhere within that 3D volume. We can use digital extraction to pull out that surface if the surface is what you're interested in. But we can also image a little bit deeply, more deeply into the tissue. When we first started this, we wanted to ask a question of how well could a light sheet microscope image in tissues compared to what we're used to. So for many years, we were doing dual-axis confocal microscopy, both point scan and line scanned, where you focus to a point and you image with a pinhole. And in this case, it's a very slow imaging technique because you have to scan point by point. Line scan, dual axis is a little faster because you collect a line of pixels. But light sheet is the fastest because you collect a whole two-dimensional image all at once, and you only have to scan in one direction to create a three-dimensional volume of data. There's another variant of light sheet in which you take a pencil beam and you scan it laterally over time to create a light sheet. That has some advantages in terms of signal-to-background ratio because you can synchronize the scanning of this pencil beam with the rolling shutter of an SCMOS camera, and it's basically a sort of like confocal imaging. You're only collecting one line at a time, and you can filter out the out-of-focus light more efficiently. So we did some Monte Carlo simulations to look at the light transport through these microscopes on a computer, through a computer code. And we found that obviously the point scan dual axis is the winner. We knew that going into this. That would give you the best contrast and depth. Line scan dual axis is what we were using for our miniature device, and we found that the light sheet microscope could get comparable performance, at least in these simulations. So that gave us some confidence that this may have some merit for looking at thick tissues. And the confocal light sheet approach would be even better, and that's a trick we can play to further increase our depth. So basically for superficial imaging of fresh tissues, for example, examining the margins of a tumor resection specimen, wide field light sheet microscopy can provide, we were estimating about 50 microns of imaging depth on fresh tissues without any clearing. This is already equivalent to 10 histology sections because most microscope slides are cut to a 5-micron thickness of tissue. And with confocal light sheet, we could probably push that to about 150 microns of depth. We also compared different technologies. There's something called Muse, which is using UV light, which has a very short penetration depth in tissue to be able to image the surface of a tissue. So this is microscopy using UV surface excitation. For 2D imaging, it's great. It's a very cheap and very fast technology, but it can only image at the surface. So in terms of depth, it's not great. But for surface imaging, it's actually quite good. On the other hand, if you look at light sheet microscopy, it's fairly well balanced in all of these axes in terms of contrast, ease of use, spatial resolution, speed of imaging, and cost. But where it really wins out is in 3D imaging. If you want to image in 3 dimensions, there's nothing like light sheet microscopy, and I think that's something that a lot of biologists have found over the past few years. This is what the system looks like. It's an inverted system, so it's just a glass plate on top. You put the tissue. So it's very flexible. Doesn't matter how large the tissue is. You place it on. We image the backside, the underside. There's a camera here. The illumination beam comes in at 45 degrees into the tissue, and it's collected at another 45 degrees. One of the challenges is the tissue to glass to air interface. So for microscopists, we know that aberrations are a big problem. We're not going to go into details, but this was actually one of the major parts we had to work on to make this work, because light just does not want to propagate well between different materials at a sharp angle like this. But by using this hemispherical solid immersion lens, we call it, we found through simulations and in practice that it allows us to mitigate a lot of these aberrations to get very clear images into tissues. And by collecting these light sheets, by scanning the tissue laterally, we're able to collect that 3-dimensional volume of data. This is what the device looks like. Fits into about 2 by 3 foot bench top, although I think we could package it further into a small box that could go into a clinical setting. Again, to scan tissues, we're scanning thin strips. The light sheet thickness is about 1 millimeter. So we scan it and back and forth in a serpentine pattern to image a large 2-dimensional area. So we get large fields of view, up to 10 by 10 centimeters now with our current device. But we also have very high resolution. We know that we can get down to micron scale, a couple micron resolution. This is called the axial response of the microscope. It tells you the section thickness. When you're imaging, it's sort of like optical sectioning thickness of the microscopes, about 3 microns for this device. This is an example of breast tissue. It's about 4 by 4 centimeters, quite a large specimen in this case, but it took us a little less than 10 minutes to acquire this image. This tissue was stained with something, a chemical known as acridine orange, which stains the cellular nuclei. So if you zoom in, you can see that these nuclei are lighting up very brightly in this breast specimen. And here's the H&E pathology. H&E stands for hematoxylineosin. The purple hematoxylin stains the nuclei, and the pink eosin stains the cytoplasmic structures. So we're getting comparable features, but it's not in 2 colors. So next, we wanted to simulate H&E, so we used a nuclear stain known as DRAC5 at 1 wavelength. It's a fluorescent dye. And then it turns out eosin fluoresces already, so we can stick with eosin, image those 2 color channels, and then false color them to create something that looks like H&E. This is an example of prostate, about 2 by 2 centimeters. If you zoom in, you can see the cellular nuclei quite clearly. So it's sort of like a Google Maps interface. You can zoom in, you can zoom out, and it gives you a lot of information, as well as image at different depths. So here we're showing just the surface, but there is some depth information, which I'll show in a second. So another application, previously I was alluding to sort of intraoperative guidance, looking at breast specimens or prostate specimens very rapidly. But there's a big application for using this for looking at core needle biopsies. And so the importance of this for pathologists is that much of the treatment of a patient depends on analyzing these core needle biopsies. If you think you have some sort of cancer, oftentimes the first step is to stick in a needle, grab some tissue out, and examine that biopsy specimen. From that, the clinician may decide whether you need surgery, radiation, chemo. It's hugely important for the treatment management of the patient. Unfortunately, there's a lot of errors in this process right now. Some of it has to do with sampling errors, a lot of inter-observer variability, which we can talk about more later. But for our device, what we're trying to do is image the entire 1 millimeter diameter core in 3 dimensions. And so what we have to do first is to use an optical clearing technique. These are chemicals that basically homogenize the refractive index profile in the tissue. And it removes the lipids, actually, so that things are more in the aqueous state. Let's see. Last time it just took a minute to get back. Let's hope for the best here. Okay, seems like it's just needs to reset itself every once in a while. Okay, so let me show you. This is an example of a core needle biopsy. There's a volume rendering on top. And we can slice and dice it in different ways. This is a full 3D data set, so we can look at it sagittally, axially, coronally, and at very different depths. What's interesting to the pathologist is a lot of these glandular structures for prostate are very useful for them to grade the tumor. For example, many of you have probably heard of the Gleason scale for prostate cancer. And it turns out that sometimes they'll see what looks like poorly formed glands, which is what they call a Gleason pattern 4. That means the patient would likely need surgery or radiation. But if you look at different depths, you can see that these poorly formed glands are actually fully formed glands if you look at different depths. That's Gleason pattern 3, and those patients do not usually have surgery. So these are critical decisions that the clinicians need to make based on doing the pathology right. And so we feel like having that 3D information could really transform and improve the art of pathology. Here's a video just of that same one small section of that biopsy. So you can see, you could fly through and see the glandular structures here. So I'll let this loop once. You'll see some glands appearing here, and you'll see that they all end up interconnecting at some point. And so some glands may look poorly formed on certain depths, but when you look at the full three-dimensional data set, you can tell that they're actually fully formed. Here's another example where we're actually using a fluorescent antibody in this case. The optical clearing technique uses an electrophoresis system, and it actually allows us to bring these antibodies into the tissue very efficiently. Usually that's a challenge. Getting antibodies to penetrate deeply into tissues is not easy, but this optical clearing actually helps us to be able to stain at the microscopic level, in this case, cytokeratin that's used by prostate pathologists to determine grading as well. Okay, so I mentioned there's some applications. Originally, the pathologists came to us because they wanted a way to triage post-operative tissues. So they bread loaf these prostates, and they have a lot of tissues to look at. They wanted a way to rapidly image the surfaces and decide which ones had tumor, in which case they would embed it and do the full pathology workup. Since then, we've realized that there are other probably better applications to look into intraoperative guidance of breast tumor resections, as well as more of a long-term project to really transform the way pathology is done with these core needle biopsies and trying to improve. Ultimately, we're trying to improve patient outcomes by getting the pathology more accurate and making sure these patients have the right treatments. So advantages are speed and cost. It's a very fast and relatively simple imaging technology where we can get comprehensive 3D pathology, very minimal sampling errors. We're imaging the whole tissue in this case. And it's non-destructive, so these tissues could be sent for further molecular assays, further testing, which is nice. Okay, so let me just acknowledge, so the light chain microscopy work is being done or led by Adam, who was a postdoc here with Brian, very great, he was a graduate student here. Now he's a postdoc with me, but a very productive member of the lab. Nick Reeder here is a pathology resident who's doing research for 1 year in my lab. And so they form a really nice team where basically they're working hand in hand, the clinician and the engineer. It's a perfect setting. And then other graduate students in my lab and postdocs. We had some help with the Sears nanoparticles early on from Brian Wilson's lab. Kinetic modeling of Sears nanoparticles with Ken Tischauer, who used to be here. And then some former collaborators at Stanford and at Sloan Kettering. I guess my time is up. Okay, maybe I should just pause and thank you for your time.
In the tissue clearing, what happens? You said it gets rid of lipids, right? What happens to the cellular membranes, or sorry, not cellular proteins like EGFR and the two?
As far as we can tell, the proteins stay intact, and that was part of what makes the optical clearing. There's many different protocols. There's Clarity. Our approach is based on Clarity. It came out of Stanford. There's Focus Clear and many other companies developing their own. But the key is that you have to make sure that you preserve as much of the structure. You don't want it to perturb things. And so I'm not an expert by any means on that chemistry, but something that they worked hard on. And so, we have to validate that, obviously, with pathology, but it seems to be fine in terms of the protein location.
What's the limit of thickness that you can image through with the light sheet microscope?
Without tissue clearing or with tissue clearing?
With tissue clearing.
So with tissue clearing, we can image through the entire 1 millimeter diameter corneal biopsy, which is nice. With fresh tissues, we're limited right now to about 50 microns typically, which is still better than pathology, being 5 micron thick. But we're trying to push that further. We're building our second generation device now to improve that a little bit. Yeah, definitely. So the machine learning and automated image interpretation is definitely a very important next step. And so we're not experts in that area, but we're trying to reach out to some data science people in Seattle and elsewhere, companies like Philips that have experience with that in the MR world or the PET world. But I definitely think that's a huge area that we are just starting to look into and would be very impactful. But right now, the pathologists still need to be in the loop, and they have to help with the development of those algorithms for sure and validating them. So this is not going to be something that rolls out in a few years. We're looking at this, and I think a lot of companies are looking at this as sort of the 10, 20-year future down the road. Can we just change the way pathology is done entirely? It's not going to be an easy path because it's so entrenched. The current processes, equipment is all set up for slides and H&E. But, in maybe our lifetimes, we might see a change.
I just have a couple questions. The endoscopic method you displayed, can you distinguish between like ulcers and edemas, or is that based on cellular density that you're able to tell if there's a tumor in the esophagus?
So the endoscopy, the spiral scanning endoscope, is a low-resolution imaging. It's a wide area, but not microscopic imaging. So the way we distinguish tumors from normal is based on their molecular expression pattern. So to the extent that these ulcers or these different conditions would or would not express certain biomarkers, then we can distinguish them. But there is a spatial resolution limit. Right now, typically we scan at a 0.5 millimeter step size. So if it's just a few cells, tumor cells, anything smaller than about 0.5 millimeter, we would be challenged to see. So co-registering, are you talking about the 3D microscopy?
Yeah, like you saw that video where you essentially pause at one slice and you can actually translate that back to one particular section that you might have taken afterwards and leave it as your 2D.
So for example, with this corneal biopsy, figuring out at what plane or in the bulk specimen, figuring out where it came from or what the tumor was located. Yeah, no, for sure. This is, with the volume rendering, we could actually cut through this 3D data set and find, whatever regions there are. And assuming that the tissue doesn't deform too much. One idea we had is to actually place the core needle biopsy in a glass tube before imaging to keep it very straight, and then maybe have some system to stain and rinse in a sort of fluidic system. But then maybe you can go back and manipulate certain regions based on the imaging.
Really interesting talk. I had a question about the microscopy for fluorescence guidance in the brain. Looking at proportion 9, once you start getting to the cellular level of proportion 9 expression, normal tissue also expresses proportion 9. So how do you kind of distinguish at that small level what is normal tissue and what is tumor?
Yeah, so that's where we really need to have some quantitative image processing, and we're just starting to work on that and figure out, are there image processing metrics where we can distinguish different grades of tumors, different cell densities, tumor cell densities, and presumably with the normal tissue, there's a difference. It's not going to be as high expression as the low or high-grade gliomas, and if we can quantify that effectively, then you can distinguish between the tissues. So the heme synthesis pathway for PP9 production is endogenous to it's a naturally occurring process. The tumors just alter it a little bit so that the PP9 accumulates more. So with that understanding, there should be more PP9 in the tumors than the normal. And so or the spatial pattern may be different. We need to explore all of that before this could be useful clinically. Yeah, that's a very good question, and you sound like a grant reviewer. That's a tough question. So depth is controversy. Breast cancer is full of controversies, we found. It's a very hot area. There's a lot of different opinions. But the recent consensus is that for some subtypes of breast tumors, invasive carcinoma, making sure that there's no tumor at the margin surface, the very surface is good enough. They call that no tumor on ink, the inked surface of that specimen. So that's where this approach would be fine. We could tell them comprehensively we're imaging the surface, which is different from pathology. With pathology, they usually cut vertical sections at different locations at the margin. So they're not comprehensively imaging the whole surface. So actually, I think this is a better approach for imaging the surface for looking at tumor on ink. But there's other types of cancers like DCIS, in situ cancers, where a lot of clinicians still want to see how deep is that tumor from the margin. And if it's within 2 millimeters or so, they still want to re-excise the patient. So maybe we can't help those patients as much. But right now, it's just so many patients, 20 to 50% of patients have to get repeat surgeries. If we can just knock that down by half, that would be pretty significant. So it's not going to be the perfect solution for all cases. But that's where we're thinking maybe we want to combine the molecular imaging with the 3D microscopy work and provide complementary information, or maybe CT or like things that people are doing here to look underneath the surface. There may be a combination of technologies.
Okay, thank you. It's pretty rare that you see a technology that you honestly I feel like would change clinical practice completely. I feel like 3D microscopy is it.
So okay, good to hear.
I'm really honored to have you come and give the talk. Thank you very much.
Okay, thank you.